Fluorescence in situ hybridization, or FISH for short, is a method used in biomedical research to detect DNA sequences using fluorescent probes. These probes hybridize with denatured sampled DNA. Using a fluorescent microscope, we can visualize the tag sequence. However, this conventional method is expensive, time-consuming, and requires experienced users. By scaling the test down to a microfluidic device, we can make the FISH process both inexpensive and efficient. The functionality of this method is made possible by different microfluidic phenomena, such as capillary action, mixing, and diffusion. Capillary action, or capillary force, is the idea that liquid molecules are attracted to the molecules on the surface of a solid because of properties of liquid such as adhesion, cohesion, and surface tension. The adhesive properties allow liquid molecules to stick to each other and the surface of the solid. Surface tension is created by the cohesive forces. In this demonstration, two napkins are placed in water, which are colored yellow and blue. One end of the napkin is placed in the colored water, while the other end sits in an empty cup between the yellow and blue cups. The colored water from both cups is transferred through the napkin and into the empty cup via adhesive and cohesive forces. This results in the green-colored cup seen between the yellow and blue cups. Another phenomena we will describe in our experiment is mixing and diffusion. Diffusion is the process of molecules traveling from a high concentration to a low concentration, which also allows for mixing of molecules, as you can see with the food coloring in water. With microscale devices, the volume decreases, which leads to an increase in the surface to area volume ratio. As a result of this larger ratio, a faster diffusion process is observed. Thus, diffusion and mixing can be controlled by the shape and size of the channel. These phenomena will be depicted by the following experiment.
In the first demonstration, a 7-inch channel is carved from a gel slab using a tool with a radius of 1/4 of an inch. Also, the two liquids are assumed to have similar viscosities to water. In microfluidic channels, conditions are vastly different. In these instances, blood has less viscosity in comparison to water. Channels are fabricated with smooth or patterned edges, and gravity has a minimal effect on the system. Due to the instruments available, the channels are not smooth or rectangular, and they will induce a gravitational effect. Lastly, the demonstration has been sped up by a factor of 1.5. Like the previous demonstration, the conditions are all the same except for the channel length. The distance here is about 14 inches. The demonstration has been sped up by a factor of 2.